Thanks, Colin. Let's make a start. I'd like to begin by acknowledging the traditional owners and custodians of the land on which we meet today, the Wurundjeri people of the Kulin Nation, and pay my respects to elders past, present and emerging, and pay my respects to any elders from other communities who may be here today. It's a great pleasure for me and surprise for me to introduce today's speaker. I only learned a few minutes ago I was doing the introduction. Len had really hoped to be here to introduce Alex, but unfortunately technology has gotten in our way. So today's speaker is Alexandra Roth Schulze. She's been a member of my lab as well as Len Harrison's lab for the last 5 years. Alex is a bioinformatician who works on microbiomes, and for the last 5 years she's been working on the Untitled study, which is a study into early onset diabetes and the environmental links associated with that, and Len co-leads that study. Prior to coming to WEHI, Alex undertook an undergraduate degree in genome sciences in Mexico before coming to Australia in 2010 to do a PhD in environmental microbiomes or metagenomics. Today she's going to talk about her work on the Untitled study. Alex.
Hi, thank you very much for the introduction, Tony. And sorry that it was last minute, but everything okay. And thank you for coming. It's nice to see people in person as well. For a long time, I've been working from home, so it's a lot of people today. So I'm going to be talking about type 1 diabetes in pregnancy and its association with the composition and function of the gut microbiome. So first, I would like to mention, can you hear me? It's something, I guess, yeah. First, I would like to mention that all of what I'm going to present is part of ENDAIA. So ENDAIA stands for Environmental Determinants of Eyelid Autoimmunity. And eyelid autoimmunity is the appearance of autoantibodies against antigens in the insulin-producing beta cells in the islets of the pancreas. These islets are regions of the pancreas that contain endocrine cells, including beta cells, which produce insulin, a hormone that controls the amount of glucose in our bloodstream at any given moment. An autoimmune response against these islets refers to the appearance of antibodies that attack and destroy the beta cells, and consequently the production of insulin is reduced and even completely suppressed. This autoimmune response precedes the onset of type 1 diabetes. So worldwide, the incidence of type 1 diabetes has increased 2 to 3-fold since 1980, including in children with lower genetic susceptibility. Therefore, in ENDAIA, the hypothesis is that gene-environment interactions during prenatal and postnatal development drive the development of eyelid autoimmunity and type 1 diabetes in children genetically at risk for type 1D. And children genetically at risk of type 1D are children with first-degree relatives suffering from type 1 diabetes. That could be either the mother, the father, or a sibling. ENDAIA is a longitudinal study from early pregnancy through early life of environment-gene interactions. So diverse samples are collected during pregnancy, at birth. Where's the cursor? I don't see it. Here, sorry. At birth, 1 week after birth, and then 3 monthly until the age of 2, and then 6 monthly until the age of 10. The initial goal was to enroll 1,400 infants at risk for developing type 1 diabetes, but with the effort of ENDAIA recruiters, we achieved already 1,500 participants. And the results I'm going to present today are from analyzing only samples from the mothers during pregnancy. I also wanted to mention that recruitments, sample collection, and research happens nationwide across Australia in all the, where you see here the blue dots. In Endaya, different molecular and cell biology aspects that can influence the development of this immune response are investigated. In order to do a comprehensive assessment of what is happening before and after the development of eyelid autoimmunity, where it goes from how the environment or exposome such as the diet, can influence the epigenome, the microbiome, and the metabolome, and how all this ultimately affects cell function and the health status. Well, each of these aspects are investigated by different members of Endaya, and I am dedicated to the microbiome. So, but what's the microbiome? Well, the microbiome is the community of bacteria, viruses, and fungi that inhabit a specific niche, such as the human gut and the genetic material within them. And today, of course, I'm going to talk about the bacteria, not fungi or viruses for now. Unlike the host genome, which is relatively constant, the microbiome is dynamic and changes with early development, environmental factors such as diet, or life events such as disease. And the human microbiome plays an important role in control of vital homeostatic mechanisms in the body, such as protection against pathogens, the synthesis of essential vitamins, immune system development, metabolite production, such as short-chain fatty acids, and modulation of the central nervous system. As part of the microbiome in Endaya, vaginal, oral, fecal, and skin samples are collected. But what I will present today is all about what we have observed in the gut microbiome. Well, pregnancy is a natural life event involving major physiological adaptations, but studies of the microbiome across pregnancy are scarce, and the findings are inconsistent. So in the first paper that I'm going to mention, because there are basically only two, in 2012, Koren et al. Compared trimester 1 to trimester 3 and observed an increase in bacterial richness and changes in structure and composition. 3 years later, Di Giulio sampled weekly and observed no differences in diversity of structure. So it's inconsistent one to each other. So pregnancy in which the mother has type 1 diabetes increases risk for mother and child, but knowledge of the gut microbiome in this context is lacking. And therefore, the aim of the study that I'm presenting now is to characterize the composition and function of the human microbiome across pregnancy in women with and without type 1 diabetes using metagenomic sequencing. For this, our experimental design consisted of 20 pregnancies, 36 in women with type 1D diabetes, and each woman gave from 1 to 3 stool samples during pregnancy, giving a total of 134 stool samples. Here, I just would like to make clear that these women have classical type 1D before pregnancy and not gestational diabetes. And indeed, women with gestational diabetes were excluded from this data set and analysis. Also, I wanted to mention that, as you can see, these are the number of samples that were collected for each trimester for this study. And in trimester 1, we only have 23 samples in comparison to 68 samples in trimester 3. And this is because early in Endaya, mothers that were, well, women that were already pregnant could enroll the study. And that meant that maybe they were already in trimester 2 or trimester 3. So, and later, as Endaya became a little bit more known, mothers started to enroll before. So that's why. And, well, these two samples were extracted and whole shotgun metagenomics was performed with the NovaSeq 6000 sequencing machine. And an analysis based on reads was performed. And that is that reads were not assembled into contigs prior to analysis. And in this analysis, we first perform a quality filter, and the sequence belonging to the human genome were filtered. And this was done with a program called NIDATA. And then subsequently, we use Metaflan2 to obtain a taxonomic profile, and Human2 to obtain functional profiles. And in functional profiles, we get metabolic pathways from the MetaSeq database, and also Keck orthology, which are enzymes. These two programs are part of the biobakery of Professor Curtis Utenhauer's group from the Department of Biostatistics at Harvard School of Public Health. And the biobakery is actually a collection of workflows for analyzing microbial communities. And it uses standardized and validated tools and parameters. And they have other types of programs that can be used, but here I only use these three. Well, when assessing the composition of a microbiome, one has to choose a unit of analysis. And these units are called taxa, and can belong to any of the levels of the taxonomic hierarchy shown here. And our analysis were done at all of these, from phylum up, down, even to strain level. That is not even shown here, but this is within species. And that and we were able to do that because we were using metagenomics. A little bit of ecology concepts that we use in microbiome analysis. Well, different aspects of the microbial communities can be measured. And one of such aspects is alpha diversity, which is defined as the total number of taxa found in a sample and how evenly distributed these taxa are in the sample. And here I'm showing a cartoon example of 3 different bacterial communities along a gradient of increasing diversity. So here the color of the bacteria represent different species. In this first community, there are only 2 species, and therefore the richness is low. And also you can observe that the community is dominated by the green bacteria, which means that it's not even. So, but then in this second community, you have now 6 different species, so the richness increased, but again you have a domination of these blue bacteria, so it's not even. But in this third community, you have the richness increased, and you can see it's more evenly distributed between the different species. So in order to determine if the diversity is different between groups, let's say diabetic versus non-diabetic women, we use generalized estimating equations. Then another measurement of, is beta diversity, which is based on the number of species and abundances shared between 2 communities. So here I'm showing an example in which I compare sample 1 with sample 2 and sample 3. This is a very simple example since clearly the community of sample 1 and sample 2 are exactly the same, and sample 3 is very different. So in order to evaluate the species that these communities share, we translate this into a table that has counts of each species in each sample. Then the Bray-Curtis coefficient is used for measuring the dissimilarity. And based on this, we can say that sample 3 is more dissimilar than sample 2 to sample 1. And Bray-Curtis can be used to cluster the samples into groups as well, using different methods such as PCOA or hierarchical clustering. And with hierarchical clustering, you can generate a cladogram like the shown above the table that tells you or denotes the relationship between the samples. And to determine if there are differences between groups of interest, we can use permutational multivariate analysis of variance. That's what we use, Permanova tests. And yeah, that's it here. Similarly to differential expression analysis of RNA-seq, Here we want to detect if the abundance of a specific taxa are significantly different between groups. Only here, rather than expression, we measure the abundance, and rather than genes, we have taxa. And therefore, it is common for people to use differential abundance expression testing tools to perform differential abundance of taxa. In this study, we use Lima and HR. In all of these analyses that we did, we control for repeated measurements, which are samples from different trimesters or different pregnancies, but that belong to the same mother, because a mother could give, well, sometimes the mothers gave samples from different pregnancies. And we also adjust for our technical variable, which is a DNA extraction batch, and for other factors that we know that can affect the microbiome. In our case, where conception age, body mass index, parity. In parity, we define it here as or 0, the woman hasn't had any previous births, or 1, the woman has had 1 or more births. And we also control for HLA type, which is the human leukocyte antigen, which are cell surface proteins responsible for the regulation of the immune system. And we know that mutations in HLA genes have been linked to type 1D, and also we know that the microbiome, actually HLA type can have an effect on which microbial community a person has. So therefore, we need to adjust for their effect in our models. And from here, I'm going to talk about the results, the actual results from the study, specifically first for the taxonomic part. So in this plot, you can see the relative abundance of the 25 most abundant species in women with and without type 1 diabetes. Each bar represents a sample. It is interesting to observe that although there is a total of 340 different species, the whole cohort, in the whole cohort, 25 species dominate. So the 25 species are the bright ones, the very bright colors, and the rest, for each bar, is the dark part at the bottom. So it's interesting that these 25 species are representing more than 50% of the sample, on average. And as mentioned before, from the two studies, one of the studies, microbiome in pregnancy, was able to detect an increase in richness during pregnancy. However, we did not detect significant differences between trimesters in our study now. Also, a previous study detected a decrease in richness in children at the time of type 1D diagnosis, but we did not detect differences between pregnant women with and without type 1 diabetes. And so in contrast to the children in that study whose microbiome was assessed at the time of type 1D diagnosis, the women in our study have already established type 1D for a long time. So this could be a reason why we don't see any differences. However, we did observe that type 1D status has a significant effect on beta diversity. And as we detected that there was an interaction between type 1D status and trimester, we had to analyze each trimester separately. And in that way, we detected that there was a significant difference at the strain, species, genus, and family level, but only in trimester 3. And the PCO plots here show the beta diversity in women with and without type 1 diabetes, red with and blue not, without. And here is the trimester 3. This is a strain level, and this is a species level. This significant interaction that I mentioned in the previous slide also meant that we had to analyze differences by trimester, separating samples from women with and without type 1 diabetes. And we were able to detect a significant difference between trimesters at the strain, species, and genus, and family levels, but only within women with diabetes. So women without diabetes didn't change, the microbiome didn't change during pregnancy. And here I'm showing a diagram that summarizes these observations about the community structure. We see that in trimester 1, there is no difference between women with and without type 1 diabetes. So we have this same community. So the microbiome remains stable from trimester 1 to trimester 2 for the two groups of diabetic and non-diabetic. And while this stability remains for non-diabetic women, we detected a change in the microbiome of diabetic women by the third trimester, which translates as well into significant differences being detected between women with and without type 1 diabetes only in trimester 3. So now, by performing differential abundance analysis, we detected significant differences in specific taxa between women with and without type 1 diabetes. Here, when a significant difference between type 1D and non-type 1D was detected across all trimesters, it's denoted with a plus sign here in the top corner. And when this difference was detected only in a specific trimester, it's denoted with an asterisk. And in here, you can see that only happens in trimester 3. So the top 3 plots here show species B. cacae, B. vulgatus, well, Bacteroides cacae, Bacteroides vulgatus, and Bacteroides uniformis. And the bottom left plot shows the Enterobacteriales order, all of which were more abundant in women with diabetes. While species belonging to the Bacteroidales order and the Bifidobacteriales order as a whole were decreased in women with type 1 diabetes. So the presence of Bacteroides cacae has previously been linked to inflammatory bowel disease, which is a group of intestinal disorders that cause inflammation of the digestive tract. So we then wanted to explore the species within these differentially abundant orders and found that most of the Enterobacteriales order is composed of Escherichia coli, which is this one, and non-classified bacteria of the Escherichia genus. And most of the Bifidobacteriales order is composed of Bifidobacterium adolescentis and Bifidobacterium longum. So we then extracted these 4 species and tested for differences in each trimester. And as you can see, E. coli was differentially abundant and more abundant in women with type 1 diabetes in trimester 3. Escherichia, the unclassified Escherichia genus was not different. But then here, for the Bifidobacteriales order, Bifidobacterium adolescentis was significantly less abundant in women with type 1 diabetes, but only in trimester 1, and no, Bifidobacterium longum was not. So products of E. coli, including lipopolysaccharide and microcin, promote intestinal inflammation, intestinal permeability, and low-grade systemic inflammation, and are implicated most clearly in the pathogenesis of inflammatory bowel diseases. While the Bifidobacterium genus members are well-known beneficial bacteria able to digest plant oligosaccharides. And a decrease in bifidobacteria has been previously associated with the development of eyelid autoimmunity or type 1 diabetes in children, including in the large longitudinal TEDdy cohort study. Bifidobacteria are also known to function in conjunction with other bacteria via cross-feeding in order to promote their health effects. And this includes, for example, producing lactate for consumption by butyrate producers to support butyrate production, or, for example, production of the biotin precursor to promote biotin production by other taxa. And also, particularly Bifidobacterium adolescentis, and this is recent, has been correlated with increased butyrate production and improve glycemic control following dietary acetate and butyrate supplementation. And this has been done in humans and is still not published. To validate some of these observations, we then performed real-time quantitative PCR on the same DNA samples from trimester 3 for species Bacteroides vulgatus and Bacteroides cacao. And we confirmed that the abundance of these bacteria is higher in women with type 1 diabetes. And furthermore, the relative abundance obtained by metagenomic sequencing and qPCR were strongly correlated. You can see here Spearman correlations of 0.74 for Bacteroides vulgatus and 0.91 for Bacteroides cacao, which is really good. So from now, I'm going to talk about the functional analysis part. We detected as well that women with and without type 1 diabetes have a significant different functional profile, but similar to before, only in trimester 3. This mirrors actually the taxonomic structure analysis. And the PCO plots here show the beta diversity for women with and without type 1 diabetes for complete pathways and for the enzymes, Keck orthology. In the differential abundance analysis, we were able to observe 6 main groups of functions that were different between women with and without type 1 diabetes. And here I'm showing one example of a functional feature for each group. So functional feature, I call it to one pathway or either a pathway or either an enzyme. So this would mean that here, actually in parentheses, on the top of each of the plots, you can see a number. This number refers to the number of functional features that were assigned to this group. And for example, for LPS production, 20 functional features were part of the LPS production pathway. Well, LPS production, or the presence of LPS actually, is more abundant in type 1 diabetes and promotes the secretion of pro-inflammatory cytokines in the host and triggers inflammation. On the other hand, we have short-chain fatty acids, the production of short-chain fatty acids, which was less abundant in women with type 1 diabetes. And we know that they are anti-inflammatory compounds, like, for example, butyrate. We also observe vitamins B6 and B12 production decrease in diabetic women. And this is of interest since mammals cannot synthesize these vitamins and must acquire them from the diet and gut organisms. So all of the B group vitamins contribute to the regulation of the immune and inflammation, and of the immunity and inflammation indeed, and their deficiency has been associated with inflammatory disorders as well. And it has been reported that it is common to see deficiency of these vitamins in diabetes. Also, we saw here the degradation of mucin produces oligosaccharides and acetate and propionate, and together these stimulate mucus production and enhance epithelial integrity to prevent gut leakiness. And also the translocation of toxins and the dietary antigens into the blood. And this enzyme was decreased in women with type 1D. Also a little bit, we don't really know now, but this vitamin K2 was increasing women with type 1 diabetes, and we've seen that a small study based on metagenomic sequencing on the gut microbiome also observed an increase in the vitamin K2, but in people with type 2 diabetes. We know that vitamin K2 is required for blood clotting and bone health, but why its synthesis by gut bacteria is increasing diabetes is unclear. And now in the next slide, I'm going to concentrate in these two examples of LPS production and short-chain fatty acids. So differentially abundant functional features were not contributed by a single species, but rather a combination of species. You can see here the species contributing to this pathway. The first plot shows the abundance of the species contributing to a pathway of the synthesis of the component of the bacterial lipopolysaccharides, which was increased in women with type 1 diabetes. And this second one is an enzyme of the pathway of the synthesis of the short-chain fatty acid butyrate. And it was decreasing in women with type 1 diabetes. So now what we did was to group the abundances of these species of each of the 6 main groups that I showed before, and into bacteria clusters. We call them bacteria clusters. And here you have the abundances in diabetic and non-diabetic women of all of the 6 bacteria clusters. And we observe a significant difference in the abundances between women with and without type 1 diabetes for each of them in some trimesters, if not all. And here you can see that there's an N on top of each of them. It denotes the number of bacteria species that form part of that specific cluster. So, for example, the LPS production bacteria cluster is formed of 19 different species of bacteria. So identifying these bacteria species could help for the purposes of biomarker or probiotics development. For example, hypothetically, if a deficiency in short-chain fatty acid producing bacteria is observed in a child with high risk at developing type 1 diabetes, perhaps this child could be supplemented with a probiotic containing bacterial species from this short-chain fatty acid production bacterial cluster, and hopefully this will prevent this child from developing a leaky gut, inflammation, and ultimately eyelid autoimmunity and type 1 diabetes. So here I'm showing all bacterial contributors to LPS production. And of interest, this includes Bacteroides caccae, B. vulgatus, and B. uniformis, which we show to be more abundant in type 1 diabetes in the differential abundant analysis. And also Escherichia coli, which was the dominant species within the Enterobacteriales order, which was also differentially more abundant in women with diabetes. And the other species could also be considered biomarker candidates, so they would be likely to be more abundant in people with type 1D. And it's interesting to observe that many of them are from the genus Bacteroides, and because members of this genus have been associated with other anti-inflammatory diseases. So since we detected in women with type 1 diabetes an increase in bacteria species which are suspected to cause inflammation, as well as an increase in the functional potential for the production of LPS, and also we detected a reduction in the production of short-chain fatty acids, we look for the evidence of gut inflammation. So for this, we measure fecal calprotectin, which is a marker of intestinal inflammation, which leads to increased epithelial permeability, and serum intestinal fatty acid binding protein, which is a marker of intestinal epithelial damage. And we found that these were significantly increased in women with type 1D. And we know that increased gut leakiness has been described in established type 1D, and recently by ourselves as well in association with gut microbiome changes in children with eyelid autoimmunity who progress to type 1 diabetes. So now, to illustrate the observations from this analysis, I adapted this diagram showing epithelial intestinal cells. Here you can see that in non-diabetic women, there is production of short-chain fatty acids by the gut microbiome. And these promoters of short-chain fatty acids include Bifidobacterium, Eubacterium, Coprococcus genera. These short-chain fatty acids, in particular butyrate, help in maintaining the tight junctions, preventing the translocation either of food, bacteria, or bacterial metabolites. And this helps maintaining homeostasis in the body. So in contrast, we observe in diabetic women a decrease in bacteria producing short-chain fatty acids or promoting the production of short-chain fatty acids, and an increase in gram-negative bacteria, like Bacteroides cacao or Bacteroides vulgatus, that produce LPS, as well as an increase in functions related to LPS production. So this LPS promotes the secretion of pro-inflammatory cytokines, which triggers inflammation. That compromises actually the tight junctions leading to gut leakiness. And LPS then translocates and ends up in the peripheral circulation, and this causes immune activation and systemic inflammation, possibly underlying pregnancy complications. And so in summary, the gut microbiome summary from my talk is that the gut microbiome changes in composition and function across pregnancy in women with type 1D. Pregnant women with type 1D exhibit a decrease in bacteria that produce anti-inflammatory mediators and essential B group vitamins, along with an increase in bacteria that produce LPS. And the changes in gut bacteria may account for the increase in markers of gut inflammation observed in women with type 1 diabetes. And we propose that a pro-inflammatory gut microbiome may contribute to the increase in maternal and fetal complications in the diabetic pregnancy, and studies are underway to address this. Our findings provide a basis for improving microbiome health by dietary or another means in pregnancy in women with type 1D, which may improve outcomes for mother and babies. Just a little bit more, a little bit of a sneak peek about what's next. So we recently sent for revision our paper already, and now that we have characterized the gut microbiome in women with and without type 1 diabetes, our next main question is, does the type 1D status of the mother has any influence on which bacteria strains are vertically transmitted to their child? So in a recent study, the microbiomes of different body sites of the mother, including oral, skin, stool, and vaginal, was characterized using metagenomic strain resolution to assess the source of transmission to the infant gut microbiome. The authors observed that colonization by maternal skin and vaginal strain is only temporal. For example, most of the vaginal species were either lost or undetectable by 1 week. And in contrast, maternal gut strains prove more persistent in the infant gut and ecologically better adapted than those acquired from other sources, which led to longer lasting colonization. Therefore, for this study, we decided to only employ metagenomic sequencing because it's the only way of getting strain resolution, and focus on stool samples. And just obviously by now, all of the children of the mothers on the study I presented are already born, and in fact we already metagenomic sequenced many of their samples. And a preliminary analysis that I performed shows nicely and clearly that the richness in the infant increases with age. In this plot you can see in the first 3 boxes the mothers, the richness in the mothers, and then from this you can see the richness in the at birth, 1 week old, and then 3 monthly until the age of 21 months. And it is also clear that the infant microbiome richness becomes more similar to the adult by this time. Probably we will need to check later points to have exactly the similar to the adult one. So, yes, here. The same for beta diversity, actually. We can see that the microbiome of the newborn, green ones here, separate from the adult ones, red, orange, and yellow. And as the infants grow older, their microbiomes become more similar to the adult microbiome. And to complete this study, we still need to sequence more samples, and we want to sequence until the first 1,000 days of life. And we will also be adding more samples from mother-infant pairs to get at least 100 100. And yeah, so with this, I would like to finish by thanking all members of the Harrison Lab at WIHI, Tony, Gordon, Alex Garham, Enrique from the Bioinformatics Division, Stephen Wilcox from Genomics Lab, Megan and Jenny and Helena from Endaya, also Curtis Utenhauer and his team, and of course the Endaya group and Endaya participants and sponsors for the contributions and efforts and help in acquiring and analyzing the data. Just for all this. Thank you very much.
We have a couple of questions in Slido, but first questions go to the audience for coming. Anna.
So I've got two questions, if I'm allowed. So one is, it's well known that the microbiome is influenced by what people eat, and they can actually change it by changing their diet.
Mm-hmm.
And I didn't see any correction for different diets in your experimental plan.
Mm-hmm. Okay. Well, actually, we know from questionnaires of what these women ate that during pregnancy that there's no big or significant difference between women with and without diabetes in what they consume. So that's why we didn't need to correct for it. Yeah, if we have seen, for example, that one group ate more fiber, we have done that, but yeah, we didn't see that. We analyzed it.
And the second question is, vitamins B6 and B12, humans have to take them up in their diet. If they don't, they get very sick. So what the bacteria, whatever the bacteria synthesize in the gut is not enough, and not enough by a large margin. They get sick and die if they don't take it up in the diet. Why do you think the vitamin B6 and B12 synthesis pathways in the microbiome matter at all?
Yeah, well, good question. We actually, what we saw was that with these questionnaires, we know that women actually take as well vitamin B supplementation, and it was no different between women with and without type 1 diabetes. That's one of the things. The other, we measure it as well from the serum, and there was no significant difference either. So we actually don't know. Maybe it's not actually doing something specifically. Maybe something related to the microbial community itself, no? Like, for example, that one bacteria is doing this vitamin, so another bacteria can use it to produce other compounds. Yeah, we would need to, I guess, do more analysis to know something like this.
Are there other questions from the audience?
Thanks for the fantastic talk. Can I ask, do you have any data on the glycemic control of the mothers and whether the women with the better glycemic control look more like a non-diabetic? And building on that, you sort of alluded to some beginnings of the thought around mechanism, but do you think the mechanism is related to glycemic control or more around autoimmunity?
More on what, sorry?
Do you think the mechanism, the difference in the microbiome is around glycemic control, or is it more around autoimmune, the fact these women have a systemic autoimmune?
We actually did a little bit of an analysis of beta diversity with those measurements, and we saw no significant difference in glycemic control. Obviously, women with and without type 1 diabetes are different, but within them, there was no significant difference. So, that was a little bit of, something that we were wondering, because obviously the glucose is part of type 1 diabetes, but we are defining type 1 diabetes as just saying all of the things, no? High sugar and the autoimmune or the immune system doing stuff. So we define it as type 1 diabetes. Yeah.
The highest voted question is, can we repeat the question?
Sorry.
So I apologise for that. I couldn't have repeated your question, Santa, but maybe you can repeat the questions when they're asked.
Sorry?
Not with Santa's, with the subsequent ones.
Okay.
Gordon has a question.
Thanks, Alex. Just following up on Anna's question about diet. So I understand your
Yeah, also the
Can you repeat the question?
yeah. So yeah, to summarize, Gordon Smythe was asking if is there a way that we can actually basically that if we can actually include the diet components as covariates in our analysis, and yeah, well, I guess yeah, we can because we have this information. I guess because we try to think about the covariates that cause most of the difference in microbiome, and we saw that in our data set this was not different between diabetic and non-diabetic women. We didn't add this because we already had many variables, you can see here, to correct for in our models, and I guess we thought since we don't see a difference, we don't want to over-adjust our model because we already have all of these 5 things. And actually the data set is not huge, it's not so big, so maybe it would be better to do it with a bigger data set. And actually we have, this data set we have a bigger data set, but with 16S. So we might be able to do something like this with this data set, with the bigger data set. I think it's just because if the data set is too small, just you are fragmenting everything too much. Yeah, I don't know.
I'm going to start alternating between Slido questions and questions from the audience. So there are a few hands, I'll get to you. There's a very highly voted for question here from Bernard. In the last part, you analysed gut microbiome of newborns up to 21 months. Do you see any difference between babies that were breastfed and those that were not?
We haven't done any analysis other than this yet, but we plan to do it, and we have the information for doing that. Yeah.
Thank you from Bernard. And from the 6 people who also voted for that question. At the back.
Thanks, Alexandra. So for some of your analysis, it was only, you observed only significance in the 3rd trimester. How much did the longer sample size contribute to that?
Yeah. Yeah, he is asking if the size of, because yeah, I showed that the number of samples that we have in the 3rd trimester is bigger than the one that we have in the 1st and the 2. Mostly in the 1st one because we have a reduced size. And how can this be contributing to us being able to detect differences in trimester 3? Yeah, actually, yeah, we did a sensitivity analysis in which we subsampled trimester 3 and trimester 2 to the same size of trimester 1, and we repeated the analysis, and we were able to detect the same differences in beta diversity and also in differential abundance. We, yeah, basically we saw the same differences. So, yeah. I mean, the best would be to get more samples from trimester 1, but we did this just to make sure that it was not the size. Yeah.
Okay, I'll get to you in a minute. The next Slido question is from Anonymous. Probiotic treatments often fail to produce a sustained change in gut microbiomes. Could diets be supplemented directly with formulations of key SCFAs instead?
Well, that's a good question, actually. Yeah, but I guess that could be an option. And the other thing that I wanted to say regarding that is that, yeah, I understand that probiotics bacteria might not establish for long periods, but they can have an effect on the microbiome even though they just pass through it. So maybe they would be doing a little bit of something, or even the genes that they share genes or something. In the process, and something can happen. But yeah, I guess supplementing with short chain fatty acids, and that's what they did in this study that I mentioned, in which they already did a trial and they were supplementing, and yeah, they saw that it was. Improvement. Mm-hmm. Yeah.
I'm wondering if you looked into any bacterial strain specificity
So you're asking if I was able to detect a specific toxin that was produced by these bacteria that were differentially abundant, right? Basically, in the because the analysis of the taxonomic part and the functional part in the beginning is separated, and then we look into the taxa that contributed to these functions. We then, when we analyze the functions, we got like a list of, I don't know, hundreds of different functions that were differentially abundant. And for now, what we were able to get, like clearly, was these 6 different functions. And of course, there are a lot more functions there that we could investigate. But yeah, it was a lot of work, and there are lots of them that still need to be checked. And we will do that as well, yeah. Thanks for the question.
Okay, question from Max Jordan. Thanks for the great talk, smiley face. What are your thoughts on the reasons for microbiome dysregulation during pregnancy in type 1 diabetes?
We have thought about it before and talked about it before. Well, I guess you could say that already with diabetes, you have like an environment of inflammation, somehow you can say, and perhaps during pregnancy, this is a little bit exacerbated. It's interesting because in the study that I talked to you about, the other one in which they actually saw differences, they saw that if they transfer the microbiome of trimester 1 into a mice, no, trimester 3 into a mice that normal mice, it becomes more inflammatory. So it's just because of the microbiome coming from a pregnant mice, right? So I don't know. It could be a lot of processes happening during pregnancy, plus the fact that the person has type 1 diabetes and already has some sort of inflammation going on there. Yeah, it's interesting because in the first trimester they were not actually different. Yeah, we think it's actually something to follow up. Yeah, to know what is happening. Yeah.
Thank you.
Yeah, thank you. Sorry.
We have a question over here.
Thank you for the good talk. It was really interesting. I was just wondering, you said that for like using these results to change the outcomes, that getting a probiotic with those specific strains would be good to give to the newborns. But do you think, considering that you showed from that other paper that was recently published that they actually get a lot of their microbiome from the mother's gut, that giving type 1 diabetic mothers during their pregnancy the probiotic would be more effective than the newborn?
That sounds like sorry, I'm gonna repeat the question. So the question is that if it would be perhaps better to give a possible probiotic developed based on this analysis to the mothers rather than the newborn, because of the study that I show, in which we see that bacteria are transmitted vertically from the mothers to the children. So actually we are in the lab. They are doing already some. They are setting up experiments with mice. And first, we want to see this transmission, and we want to see if actually there's transmission of this specific bacteria from the mother to the infant's microbiome. So that's another option, of course. Yeah, that once you know that a mother has type 1 diabetes, or the children has a risk at developing type 1 diabetes. The mother is the one. Yeah. And then
another Slido question. Let's see. Is there a similar study of gut flora for patients with inflammatory bowel disease?
Is there? Sorry.
Are there similar studies for patients with inflammatory bowel disease?
Yeah, I guess there's a lot. all the time when I find something is about inflammatory bowel disease, and it's a lot to do with bacteroides, as I mentioned, that are increased in people with inflammatory bowel disease. Yeah.
The follow-up part of the question was, have you correlated it with your data? And you've sort of just answered that.
Ah, yeah.
Are there any other questions from the audience? No, okay.
With that, could we thank Alex for a wonderful talk?
Thank you.